Okay, so first question that we've got loaded here is study the diagram below, and it is the nuclear membrane, and immediately you see this. What do you think is happening here? It is protein synthesis. Now, protein, you know what protein is, and synthesis, to make. All your cells, your hormones, your antibodies, your enzymes are made of protein. So we have to be able to make protein, but our proteins are special because the recipe for our specific proteins, each individual human being on this planet, the recipe for your proteins for your body are made, and the recipes are written in your DNA. Okay, that's why no two people on this planet, unless they're identical twins, have the same DNA. Okay, so we have the nuclear membrane. In here, inside the nucleus, okay, we have our DNA. And remember, DNA is a double strand, so there's the one strand, there's the other strand. And we've got nitrogenous bases all the way down. So we've got our phosphate, and then a pentose sugar, and then your nitrogenous base sits this side. And what do we have in DNA? We have the weak hydrogen bonds that bond the two strands together normally. And it's twisted because it's a double helix. Okay, so what they're doing here is they tell us, okay, we've got number one. So this would be our DNA, and it would be the template strand. Okay, so there's our template strand. And we can see there guanine, cytosine, adenine, adenine, cytosine, adenine, thymine, 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 guanine, cytosine, guanine. Okay, now, something I want you to take note of, anything you do with regard to protein synthesis, you are going to do the following. You're going to say adenine will always bond with thymine in DNA and in uracil in RNA. Okay? So that's the difference between DNA and RNA is thymine versus uracil. If it's got uracil, we know it's an RNA molecule. Okay, then guanine to cytosine, always. It always, adenine to thymine or adenine to uracil and guanine to cytosine. Always, always, always. A to T, G to C, done. And you write that at the top so that you do not mess it up. You must know that that's what's going to happen. Okay, now, if we look here, this, let me get my blue. This is going to be your transfer RNA. And transfer RNA floats around here in the cytoplasm. Okay? And its job is to pick up amino acids. And that's the process of translation. So it goes and it finds amino acids that fit its anticodon, which is very specific. And this would be your little amino acid. So what I'm doing is I'm giving you every possible label that you can get in a diagram like this, okay? So A is going to be in the nucleus. B will be your cytoplasm. Okay? The process that's going to take place here is going to be translation. And your process that takes place here is transcription. So transcription is when DNA makes RNA. And that's exactly what the DNA does here. The DNA makes this one, which is your messenger RNA. Okay? So here's your messenger RNA. You have your complementary bases all the way down, and then it goes zip, and it comes here to the ribosome. So it tells you that's the ribosome, this thing that looks like a fat 8. And 3 is going to be a polypeptide chain. Okay, or you can say protein, but it's not quite a protein. A protein is a whole bunch of polypeptide chains together. Polypeptide chain is the chain that makes the protein. All right, 5 would be an amino acid, which is silly because I already labeled it there. Okay, and number 2 is your anticodon. I've already told you that, anticodon. Okay, so let's just take it off here. So let me take it off there, and I'm going to take that away there because you've already got it. Otherwise, you're not going to have enough space for everything else. Alrighty, now let's look at our questions. Okay. So it says, name the process. Well, it's protein synthesis. Okay, now, when the cell does process B occur, we've already said the cytoplasm. Okay. Identify the following molecules, 1 and 3. Well, we know 3 is a polypeptide chain. Okay. And 1 was a DNA strand. Okay, not just DNA, a DNA strand. It's only one strand of the DNA. Okay, now, if we look at this, I'm going to say, let me just put it here, DNA strand. Okay, 3 is a poly, means many, peptide, lots of peptides that join amino acids, peptide chain. Okay, give the correct base sequence to replace numbers 6, 7, and 8. So let's see where numbers 6, 7, and 8. Here's 6, 7, and 8. So we go back to the original DNA strand. And when we're looking at the DNA strand, we're going to go, so this is 1, so G to C, C to G, G to C. T will go to A. A will not go to T, it goes to U because we are talking about RNA. Okay? C to G, A to U, A to U, G, C to G, and G to C. All right, so those are all that's our chain. And if we look here, we've got C, we've got G, we've got C, we've got our AAA, and then we've got UGU. So this would be UGU. And then there we go, UGC, UGC. So everything's correct. All right, so they wanted to know the sequence is UGU. Write down the anticodon for label 2. Now, if we look at label 2 is going to come in here for UGC. So if it's coming for UGC, that's your anticodon, it's your transfer RNA. It's opposite to your messenger RNA, which is a codon. Okay, it is complementary. So if we have U, it's going to be A, and G will be C, and C will be G, and there you go. There is your anticodon. Very, very easy, A, C, G. I want to show you this quickly. Mind you, we go here. Okay, let me just get a different color. All right. If we look at this, this gives us the anticodons for transferred RNA. Okay, and there are our amino acids that are going to form. But I want to show you this. I'm going to make this here, this column, messenger RNA. And then I'm going to put this column here. I'm going to make this one DNA. Okay? So, your messenger RNA is going to be it's going to be opposite to, or complementary rather, to UUU. So, what is going to be complementary to UUU? Remember, A to T or U and G to C. Those are your complementary bases. And if it's RNA, then it will have uracil. If it is DNA, it will have thymine. So if it is UUU, what's complementary? It's going to be A. What is complementary to ACA? It's going to be UGU. And complementary to GCA, it's going to be CGU. And G is C, UU is going to be AA. And CC is GG, and A is going to be U. Complementary AAA is going to be U. All right, so these are going to be your messenger RNA codons. And they are going to be complementary to your transfer RNA. Now, if we look at our DNA and our RNA, our messenger RNA, we're going to have another set of complementary here. And remember, this was complementary. So AAA is going to be UUU. Oh, sorry, not UUU, TTT. It's going to be TTT. Why? Because remember, DNA has thymine and RNA has uracil. So this is going to be UGU is going to be ACA. And CGU, we're going to have GCA. And CAA will be GTT. Why? Remember, thymine versus uracil. Okay, and then GGU will be CCA, and AAA will be, UUU will be AAA. Now I want you to look at this. DNA to messenger RNA is complementary. Messenger RNA to transfer RNA is complementary. But DNA and transfer RNA are very similar. Why? Because if you replace the thymine with uracil, you have transfer RNA. And if you replace the uracil and transfer RNA to thymine, then you have DNA. So look, TTT, UUU, ACA, ACA, they're the same. GCA, GCA, same. All right, I need for you to understand that. So you've got your DNA, always has thymine. Your messenger RNA codes your DNA, so it's complementary. Your messenger RNA and your transfer RNA are complementary. So if you've got complementary and complementary, it means that your DNA and your transfer RNA are the same, except DNA will have thymine and transfer RNA will have uracil. And guys, if you know that, you can never ever get anything wrong here. You'll get full marks every single time. So now it says, what is the DNA base triplet for glycine? So they want the DNA base triplet. So we look here for glycine. The DNA base triplet is going to be CCA. Why? Because there it is and there it is. CCA, CCA. Because your transfer RNA is equal to your DNA, except for the thymine and the uracil. Alrighty, now, name the amino acid found at 5. So if we look at the amino acid found at 5, this amino acid is going to be AAA, alright? At least it's AAA on the RNA, mRNA. It's going to be UUU on the transfer RNA, which means it's going to be TTT, and for the DNA, and that tells us that it is going to be lysine. All right, there it is, TTT, UUU, lysine. Then explain how the composition of the protein molecule would change if the base sequence for the second codon on molecule 4 was UUU instead of AAA. So looking at molecule 4, that is your messenger RNA. So if your codon was UUU instead of AAA, what are you going to end up with? Well, look at this. If it is UUU instead of AAA, it means that this is on your messenger RNA. So messenger RNA is UUU instead of AAA, what's going to happen? Your anticodon, okay? Remember your anticodon on your tRNA, your anticodon will be my brain, AAA. Okay, so UUU, your transfer RNA is going to be AAA. So what happens then? What's the next thing? Therefore, the amino acid will be, and all you do is you look at your table. So remember, it was supposed to be lysine, UUU or TTT. So you wanted lysine, but now you're not getting lysine. You're getting AAA. And what is AAA? AAA is phenylalanine, and it's a different amino acid. It's not lysine, it's phenylalanine. So will therefore be, and you just must spell it right, phenylalanine. Okay? And what's going to happen then, instead of lysine, and therefore be a different protein. You're not going to make the same protein. People, it's like if I want to make muffins, all right, or I want to bake a cake, and I use mealie meal instead of flour, it's going to have a completely different texture, and I'm not sure if it's going to be light and fluffy and bake as well as flour would. Or if I want to make mealie crumpets and I use flour, it's going to have a completely different consistency. It's not even like adding a bit of chocolate to make a chocolate cake versus vanilla cake. It changes the entire structure of the protein. And when you change the structure of the protein, you change the function of the protein. And that is not good. So you don't want to change things. Whatever's on the DNA is written onto the messenger RNA by transcription. And then the messenger RNA, because remember the DNA can't leave the nucleus. So the messenger RNA goes bitty, 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 and it goes into the cytoplasm. When it gets to the cytoplasm, it goes to the ribosome, which is where the protein factory is. Okay, it's the site for protein synthesis. And your messenger RNA goes and sits right on the ribosome with its codon. And then what happens? The transfer RNA floating around, and it has an anticodon. So it only picks up certain amino acids that have that combination of nitrogenous bases, those three. It's a triplet base. So they then would say, if the messenger RNA is UGU, then the transfer RNA is going to bring an amino acid cysteine with ACA. And that's exactly the same as the DNA. So that is how it works. That's translation. Transcription, DNA makes RNA. Translation, RNA makes proteins. Okay.